Thank you for the invitation and introduction. Today I'll be giving a brief overview of our abstract that is focused on off-the-shelf CAR T cells. I'm employed at Fate Therapeutics. So pioneering work have really demonstrated that CD19 CAR T cells can effectively target and eliminate B-cell lymphoma and leukemia. This pioneering work has led to the recent approval of 2 key autologous CAR T products as named here. All the remarkable effects have been seen. The manufacturing of autologous CAR T therapy is impaired by several challenges, including the starting material, which comes from a patient that has cancer, and is a single-dose paradigm. In what is dubbed Gen 1.1, allogeneic CAR T therapy begins to address some of these challenges by starting with healthy starting material and creates a handful of doses. However, many of the challenges still remain. Our off-the-shelf vision starts with an iPSC, a pluripotent cell that has a unique ability of unlimited self-renewal and capacity to differentiate into 200 cell types. Using this cell, we can effectively place multiple attributes into a cell at the single cell level and create a master cell bank. This master cell bank, rising from a single cell iPSC, that's a clonal population, can be plugged into our proprietary differentiation and expansion strategy to make mass number of T cells that are capable of multi-dose applications and can be basically begin to address many of the challenges as discussed earlier, including creation of a homogeneous product, a one-time reprogramming strategy and engineering strategy, a cost-effective process, and a starting material that is from a healthy starting source. Briefly, at a one-time editing event, we basically put in the desired attributes into the iPSC. At the clonal level, we'll look for the attributes that we want. And as you can see here in the middle with the gel apheresis picture, we have now identified clonal populations that have the CAR T knocked into the track locus, while it has the TCR fully disrupted through biolytic disruption. And not only we see this at the gel electrophoresis image, but also is confirmed with sequencing. This precisely edited cell can now be differentiated and expanded into a homogeneous product, as shown in the right-hand figure. As you can see, the product is pure for CAR expression and has complete elimination of TCR. So as we've have been told through several clinical trials that even few cells expressing TCR can elicit GVHD. So this pure product is now safe and highly efficacious. Using this precisely engineered cell that's been made into a master cell bank, we can now take a vial from that bank and through a stage-specific process differentiate the iPSCs with the genetic attributes into a hematopoietic cell and subsequently into a T cell in a process that scales to numbers that basically address large quantities of doses. So you can see here that one manufacturing process starting with a very small quantity of iPSCs can produce 1 × 10⁻11 cells, resulting in hundreds to thousands of doses. And because this process starts with the same vial and goes through the same manufacturing process, you can do this time and time and create the same product. FT819, as I mentioned, has CAR inserted in the track locus and has TCR disruption, biolytic disruption, so it's completely lacking TCR expression. And this product is responsive, cytotoxic, and specific. As you can see here, FT819, similar to primary CAR T, when induced, expresses TNF alpha and interferon gamma. More importantly, when placed into an anti-tumor killing assay, you can see that the product is uniform and consistent in its ability to specifically target the antigen. And as you can also see, when the cells are introduced, the FT819 is introduced into a CD19 negative population, it does not have any killing, even though through the dose escalation, you can see based on the effective target ratio, I hope you can appreciate the difference between ability to target CD19 cells and the lack of in the CD19 negative population. In addition to having a CAR into the TRAC locus and biallelic TCR disruption, FT819 also contains CD16 Fc receptor as a second target modality. This target modality can be used to mitigate antigen escape through ADCC. So this product containing CAR19 and CD16, again, is made at the single cell level, so you can see that the product, FT819, is pure for CAR19 expression and CD16 expression. When applied into an experiment where, in addition to having a pure population of CD19 cells, we also have a pure population of CD19 negative cells that are still CD20 positive, and a mixture of population of CD19 positive and CD19 negative cells, you can see that in scenarios in green and blue where CAR19 cannot target the majority of the population, ADCC helps get rid of a lot of the cells that are still cancer cells but are CD19 negative. So in conclusion, I hope that I have shown you that CAR T cells can be robustly manufactured from a multi-point engineered master iPSC cell line. The master iPSC is self-renewing and can be used repeatedly without sourcing new donor material or re-engineering. The single manufacturing campaign yields large quantities of CAR T cells and can be repeated in the same exact manner. We are developing FT819, an off-the-shelf T cell therapy which is completely lacks TCR expression, has controlled CAR19 expression through the TRAC insertion strategy, and has CD16 expression for ADCC. FT819 is an off-the-shelf CAR T cell therapy with the potential to not only improve safety and efficacy, but also address antigen escape, broaden patient accessibility, and reduce cost of manufacturing, and really begin to make this available for the masses. Finally, I'd like to thank Fate Therapeutics, MSK, CC, and especially Dr. Michelle Satterline, who not only has led the R&D activities of FT819, but also has pioneered CAR19 T cell therapy and has given a new hope to cancer patients. Thank you.